This is Bob Cox again. Now, in this 8th talk, I'm going to talk about pre-processing steps that come before regression. So, regression is really the final step in the time series analysis of the individual, especially in task studies. Before regression, the data sets are processed in a lot of ways to clean up the data, to make the data more like what we wish it was rather than what it really is. So for the different kind of experimental paradigms, task-based, resting, naturalistic, the pre-processing doesn't really vary a lot, but the main difference is the attitude towards what you get from the regression step. In tasks, the betas for the regressors of interest are the things that are of value. They tell you how much there was of fold effect in each voxel. Whereas the resting state and the naturalistic, we don't have task regressors usually, so the residuals are the output of interest. Because there are no regressors of interest, there are regressors of no interest, which are hopefully there to remove contamination from the data. And so what's left afterwards is data that was hopefully purified in some sense. So this is our recommended resting state pre-processing steps, and really pretty much the same thing applies to all other kind of experimental paradigms. The first step is very simple in the concept. It's just despiking. Sometimes you see data that has spikes in it, and you don't want these spikes because imagine regression is a least squares operation. That is, a fluctuation that is 10 times bigger counts 100 times as much because it's squared. So these things here that stick up like 5, 6, 7 standard deviations, they can completely contaminate the results. So the despiking is a relatively simple operation and is carried out and produces results like this. Problem, if not solved, at least ameliorated. Slice timing correction is needed because most functional MRI techniques don't acquire snapshot volumes in a single acquisition. Instead, 2D slices are acquired at different times, and then they're assembled into a 3D volume, and then the whole process is repeated over and over again every couple of seconds. But the result is that 2 slices, which might only be a couple millimeters apart, may be acquired half a second or a second apart. And so the same physiological effect will show up slightly differently, being due to measured at slightly different times. And so they'll be slightly less correlated, or they'll be slightly less identical than they should be in the data that we wish we had. So one solution, the most common in FMRI, is to interpolate through time to some common reference point before calculating the regression. This is not perfect because, of course, we're also interpolating noise, not just the bold signal. But it is the most commonly adopted solution because there's not a very obvious other good solution. Once we have that done, then we have volumes that are supposedly been shifted in time to the same origin. Then we want to shift them in space so they're all aligned. That's step 3. We have this sequence of volumes in time, and we do the alignment through time. And this is really important in these naturalistic or resting state fMRI because the bold effect is smaller than focal tasks. And so good compensation for subject head motion is critical. Step 4, the alignment of VPI with the anatomy, is clearly needed simply for the purpose of assigning results to brain regions. And step 5 is needed for aligning subjects to a common reference space so that we can do contrasts and averaging across groups and within groups. Now, it sounds like what we do is we align in step 3 all the brain volumes together. Now the EPI volumes are all lined up. Then we move them to be aligned with the subject anatomy, and then we move them again to be aligned with the MNI template, say. That's not really true. What we do is we calculate all those transformations needed to do that, and then we move them all in one big transformation at the end, combining steps 3, 4, and 5 into one operation. That's why they're all on one slide here. That's to avoid moving the data around needlessly a number of times, which can itself produce some artifacts. Step 6 is something we haven't discussed, and that is before, but that is creating extra regressors of no interest using the fMRI data itself. And the purpose is to find time series fluctuations that are not bold signals, so that we can then try to remove that components from the data as much as possible, so they won't interfere with the functional MRI analysis that comes after. So common choices include the average of all the white matter time series. Say you have 20,000 voxels in white matter. We don't expect very much bold signal in white matter because white matter has a lot less blood than gray matter. So average across those and use that as a regressor of no interest. I remember this being introduced sometime in the mid to late 1990s. A somewhat more sophisticated version, introduced a little later, uses several principal components of all those white matter time series. This is called the CompCore method, and using several principal components is supposed to better represent the different kinds of fluctuations that come in the white matter, again, all thought to be artifact, and therefore give a better set of regressors of no interest. Something that is controversial, which is why there's a frowny face at the end of it, is to use the average global brain time signal. That is, we have create a brain mask, average at every time point, all the numbers in that brain mask, do that for every time point. Now we have the global signal, GS, and use that as a regressor of no interest. In AFNI, you can do this. It will give you a lot of power, but we don't like this ourselves because you're using gray matter to find signals of no interest, but gray matters are where the signals of interest come from. We feel that this biases the results in ways that are essentially uncontrollable. Another possibility, of course, is to also use the signal from CSF, which is also thought to be essentially all artifact in noise. Another approach to get white matter signals is used only in AFNI because this approach produces a voxel-wise regressor. So this is a cartoon diagram of a brain segment. The squares in white are supposed to be white matter, and we actually mean by white matter here the eroded white matter, so that we take the white matter mask of voxels and erode it one voxel away from the gray matter so that we're not including any gray matter signals. And then the voxels in blue are intended to represent gray matter. The voxel in the center, say, we want a regressor of no interest for that. Instead of averaging over all the white matter or taking a few components over all of it, but then what we do is we take just an average over a region just around that voxel, the one in red. We just average over all the white matter voxels inside a 25 millimeter radius, say, at each time point, average across, can do that across all the time points, and we have a regressor for that voxel. For a voxel over here, we have a slightly different sphere drawn around it with a slightly different set of white matter voxels inside, so we get a slightly different regressor of no interest. This is done in AFNI, so this is a place where the X matrix is pretty much the same for every voxel except for this one column, which will be different for every voxel. In AFNI, we can do this because the programs that are written for regression are written to be very efficient, and so it makes the analysis still feasible to carry out even on hundreds of thousands of voxels to have a different matrix in every voxel. Then we come to spatial blurring. This is usually done in task fMRI, and it is important for resting state or naturalistic fMRI since the bold signal fluctuations in these undirected situations are smaller than for focal tasks. And it's also practically useful in group studies so that errors in intersubject alignment can be allowed for. You can't align two different people's brains very accurately, within a few millimeters if you're lucky. And so by blurring the results from each subject somewhat, then you can make sure that regions that are close to each other but not right on top of each other because of bad alignment will tend to overlap better, and you can get more reasonable results. Averaging locally tends to cancel the noise. From voxel to voxel, the noise will tend to cancel out and add up what are called coherent or similar looking signals. It is important to blur after tissue-based signal extraction, so you don't blur the white matter signals into the gray matter and the gray matter signals into the white matter, and then take out white matter signals, because now the white matter signals would have gray matter signals in them. I mention this point only because some people have done it wrong in the past. So here's an example of the effects of blurring on resting state correlation. If you imagine putting a voxel seed down at the intersection of the crosshairs and correlating that voxel time series in fMRI data with every other voxel in the brain and making a color map of the correlation coefficient, you see that if the 0 millimeters blur, then you see basically the vein in the sulcus there that is highly correlated, and then on the opposite side of the brain you see a little bit of more hot red stuff with a lot of high correlation, but you have to actually look pretty hard to see it. If we blur it a little more each time, 1 millimeter, and then go from 0 to 2 millimeters, 0 to 3, and so on, then at about 3 millimeters, you can see there's a fair amount of long-distance correlation in the brain. This is what people look for in resting state. They look for long-distance similarities in maps. There's no task in this study. The person just lies in the metal tube and thinks whatever thoughts they think. And then we look for parts of the brain that were going up and down in synchrony. So in synchrony with this location where the crosshairs are, with 3 millimeters of blurring, we start to see long distance correlations. And we go to 3, 4, 5, 6, 7, we see a lot of long distance correlations. And 5, 6, 7 are commonly used in this field because to get this strong effect that you can see here, and also when you go to group analysis so that you can get good overlap between subjects. Now we come to the regression, and I'm particularly going to talk here about nuisance regression. The nuisances include the motion parameters, which was discussed in a previous talk. Another thing you can do is the motion parameter time differences. That is, the difference between the motion estimate now and the motion estimate 1 TR ago, the derivative is sometimes called. This is how fast the subject's head is moving, not just how much it moved. White matter signals, which we just discussed. If you have the platysmal graph, the chest belt, you might have a measured respiration signal, and so on. In resting state, naturalistic fMRI, there's no task model components, so all we really care about is the nuisance components, and we need as many nuisance components as are necessary to do good purification of the data, you might say. Another operation that's sometimes used in resting state fMRI is called band passing, which is a technical term introduced in signal analysis for telephony about a century ago. And it involves taking all the frequency components from the data except those in a specific band or range. So we pass through certain bands of frequencies and we suppress the other frequencies from the data. If you're not an engineer and you haven't studied this, the units of frequency are called hertz, named after the famous German physicist Heinrich Hertz, the first person to detect radio waves. And the unit of a hertz is a cycle per second. A cycle is one full sine wave up and down, peak to peak, as in the picture. 1 hertz is 1 cycle per second. 1/100th of a hertz is 100th of a cycle per second, which is to say 1 cycle in 100 seconds. And the opposite, 100 hertz is 100 cycles per second, which means 1 cycle in 0.01 seconds. So a larger frequency means things are happening faster, and a lower frequency means that things are happening slower. In resting state fMRI, it's very common to band pass out all frequencies higher than 0.1 hertz, that is the stuff that wiggles around faster than 10 seconds or so, and all frequencies that are smaller than 0.01 hertz, which is to say we're keeping data fluctuations that occur in the 10 to 100 second time scale. Things that are faster than 10 seconds or slower than 100 seconds are taken out. The idea is that the fast stuff isn't like the bold effect, which, as we've seen, goes up and down slowly, so it should be removed before the analysis of the residuals. The reason for getting rid of the slow stuff is that there can be bold effect at those frequencies, slow learning, for example, or slow adaptations, but the problem is that there's a lot of subject head drift and stuff which isn't fully compensated for by motion regression will still be present, and we just don't trust the results at these low frequencies, at these very long time scales. The idea of throwing away all frequencies faster than 10 time scale 10 seconds of higher frequencies higher than 0.1 hertz is controversial. There's a fair amount of evidence that neurally relevant fluctuations occur at least up to 0.2 hertz, the 5-second time scale. And so that is generally in AFNI, we don't recommend that people bandpass their data if they have the unusual kind of TR of 2 seconds or so. But here's what happens if you do bandpassing. Here's a voxel on the left, real voxel data time series, and then if it gets bandpassed in this canonical range 0.01 to 0.1, then you see the result on the right. The bandpass removes that slow drift upwards, and it reduces these rapid fluctuations, but leaves the medium-sized fluctuations. And there's a script in the AFNI website that goes with this presentation, which shows you how to run a bandpass analysis like this on data. Another thing that's common is to throw out or censor bad time points. Sometimes people call this scrubbing the data, so that they aren't used in the analysis. In this case, done in task and resting state. Bad, what is bad? Too much motion is an obvious thing, or the volume just has too many outlier data points, or whatever criterion you like. Those are the two criterion we generally use in AFNI Proc Pi, where an outlier data point is described as something that's just, it's not quite a spike, but it's a little bit bigger than it seems to be, and then that's okay, but if a volume has a lot of those, it's suspect. So it is important to censor the bad time points out before or during the nuisance regression and not later after that, because if you censor them out later, then it'll affect the regression results. And obviously this makes no sense when I'm talking about task. When I'm talking about resting state, you could imagine saying, I'll just do all the nuisance regression, and then later I'll just throw out the time points I don't want to do the interregional correlation analysis. That's a wrong way to do it, because the bad data will come in and affect the regression results and contaminate the results, even if you think you've removed it later. In AFNI, censoring is done by literally removing the offending data time point from the analysis, both in the matrix and data. An alternative is to add an extra regressor, which is 0, except at the time point to be killed, where it gets set, that extra regressor is 1. So you have a column in the matrix that's 0, 0, 0, 0, 1, 0, 0, 0, 0, 0. And if you have 10 time points to be censored, you add 10 such columns. This is the way SPM and FSL do it. These things are mathematically equivalent, removing the data or putting in the 0, 1 regressors. In AFNI, this nuisance regression, the bandpass sensing, and the censoring are all done in the same program, 3DT Project. This program only computes residuals, as I mentioned in the previous talk. And this program allows for voxel-specific regressors, as does 3D RemelFit. 3DT Project, though, is a lot faster than 3D RemelFit, since it doesn't have to compute the betas or statistics. It does one thing, and it does it quickly. How does AFNI know which program to use? It's simple. If you don't give it any tasks, then you're not doing task-based analysis, and so it uses 3D project, otherwise 3D Rammelfit. I do want to mention something that has come up in the past and hopefully will never happen again in the future. People have done these two steps like this. They bandpass the data. That's one concept, and then the other one is they regress the nuisance regressors from the data after bandpassing. I have a lot of opinions about FMRI. This isn't an opinion though. This is wrong. And it's not bad. Things that are bad. When I say things are bad, that means that it's my opinion. But when I say it's wrong, it's objectively wrong. Since nuisance regressors will contain some of the rejected frequency components, say they take some of the white matter signals, those will have some low frequency and high frequency stuff. When you finally apply the regression after the bandpassing, that some of those will leak back into the results, and that will provide long distance correlations that don't exist. Now there are ways around that to do it in 2 steps, but it's just easier to do it in 1 step and then you don't have to think about it. These reasons are why 3D T-project was written. That plus the speed reason, of course. So again, here's the flow chart of what happens. And in AFNI these are implemented with a script that the command line script that has all the options that let you control how each of those things work. This is adapted from example 9b in the AFNI help, which is something that is definitely worth reading and skimming through. The examples have a lot of use cases for different setups, simple setups, complicated task setups, resting state with an ADA-core, and so on. So in particular note is the list of blocks here. These are the processing blocks as described in the program and as described in this talk. Despiking, time shifting, aligning, transformation to a template space, which used to always be Telleract, but is now many things. Ball reg, the time series volume registration, the blurring, creating masks, the regression time step. So just options here: set blur size, the amount of blurring to use an ADA-core, how much motion to censor, how bad a volume has to be before it gets tossed out as being too many outliers. Here's the bandpass here set from 0.01 to 0.2, and here the motion types. This demean means here the motion regressors with their means removed so that their average is 0. And deriv means also take the time derivative of the motion regressors, as I mentioned earlier. So we have 6 motion regressors from 6 ways a body can move rigidly in space, and we'd have 6 more regressors from the derivatives. And then these are various options for what it does after the regression analysis. So thank you. Stay tuned for more talks that are more practical about actually running AFNI to get results.